So welcome to this seminar series on breakthroughs in fluorescent imaging. In this third section, I'd like to talk about super-resolution imaging using photoactivatable fluorescent proteins. This approach, a super-resolution imaging, allows us to visualize the behavior of individual molecules, which fluorescent proteins with conventional diffraction-limited imaging cannot allow you to look at. Instead, you're looking at ensembles of proteins. Now, to get a framework for what we're talking about with super-resolution imaging, I have a little diagram here illustrating the spatial scale that fluorescence microscopy in its conventional format enables us to experience or visualize. So conventional fluorescent microscopy has a lateral dimensions of anywhere between 400 to tens of microns in imaging capability. But many molecular structures within cells are well below 400 nanometers in size. And so it's been a challenge of biologists to image in this nanometric region. And this is the region that we call super-resolution imaging. Anything from just a couple nanometers up to about 200 or 400 nanometers in size is in the realm of super-resolution imaging. Now, why has there been a problem for imaging objects as small as things like GFP, which is about 2.5 nanometers in diameter? Well, the problem is the diffraction limit of light. If we image a single molecule of GFP through a microscope, what one sees is not a single 2.5 nanometer spot. Instead, what you see is a very large blurry spot called the microscope point spread function that is 100 times the size of the single molecule of GFP. Now, if we have a specimen that's expressing many molecules that are all within 200 nanometers, which is the diameter of this point spread function of a single molecule, you can easily see how you'd have no ability to define how individual molecules are distributed within this blurry spot. And as a consequence, you have no insight into how molecules are distributed in nanometric structures that make up the vast majority of molecular machines within cells. Well, there's one way to get around this diffraction limit using an interesting approach, which is the recognition that if one looks at a single molecule, the microscope point spread function, or blurry spot, has a Gaussian spread of distribution when you look at it in X and Y. So if you are imaging a single fluorescent protein, you can, with fairly good accuracy, determine where the molecule must exist within this PSF by simply applying a 2D Gaussian least squares fit to determine the centroid of this Gaussian. When one does that, you can, with very good accuracy, determine where the molecule is situated within that blurry spot. Now, this approach of fitting the blurry point spread function exhibited by fluorophores has been used to very good avail at determining how single molecules move and behave when they are expressed in vitro. And this is just illustrated in this diagram right here and up here in a little cartoon fashion, where researchers looked at the behavior of individual motor proteins, in this case myosin, moving on an actin filament. And what you can see is the Gaussian point spread function given off by the single myosin molecule can be fit with very good accuracy to determine the nanometric steps that this motor takes in time. This is possible because of the ability to fit these blurry spots given off by these fluorophores in the way that I have told you. Now, the problem comes if you're interested in looking at more than one molecule in a dense population. And this is what's typical for how molecules are distributed within cells. They are densely packed within cells. This approach of fitting individual fluorescent probes using this kind of Gaussian fitting system falls apart under these conditions when you have many molecules, because you don't know whether in any particular PSF, which it has a radius of anywhere between 200 to 400 nanometers, whether there's a single molecule or hundreds of molecules. So your localization is essentially lost through this approach. So one way to get around this problem where the point spread function, the blurry distribution of this point spread function, masks the distribution of hundreds of different molecules, is the use of photoactivatable fluorescent proteins. These proteins, as I mentioned in the previous talks, switch on from a dark to a light state. What that allows you to do is to switch on molecules one at a time and to potentially build the distribution of a dense population of molecules. And this is the basis of a new super-resolution approach called Photo-Activated Localization Microscopy. Now, in this technique, which typically uses fixed cells that are not moving, a specimen that houses thousands of photoactivatable fluorescent protein, whose distribution you'd like to know, is switched on with very low light. The low light flips on a small subset of the overall population of molecules randomly. And so the probability that two or more of these molecules that switch on have their blurry spots overlapping is quite small. So when you switch on a small subset with this low light illumination, you can then fit the PSFs using the Gaussian fitting algorithm to determine with very high precision where those two molecules, in this case, that you've switched on, are distributed. Now, once you've done that, you can photobleach these molecules and then photoactivate another small subset of molecules, again using very low light, which just randomly flips on a subset of these molecules. Now, because the specimen is fixed, you can now determine where these molecules are distributed in the structure that you're looking at using the fitting algorithm that I mentioned. After fitting these molecules, you can do another bleach and photoactivation to switch on another population in this overall population. And you can do this over and over again, thousands of times. And after you've collected the distributions of all these molecules, you can then merge them into one big super-resolution map showing how all of the molecules are distributed within the fixed specimen. Now, this movie sequence here just illustrates how one can go about acquiring data for this type of super-resolution analysis. So this is a thin section through lysosomes within cells that are expressing a photoactivatable tagged lysosomal membrane protein. To the right is the raw image where you can see how we're photoactivating the molecules that are distributed on the membranes of these lysosomes. This is the summed distribution of all of those molecules. And here is the PALM image that's being constructed as we photoactivate individual molecules in that thin section. And with time, you can see we can build the super-resolution image of all the molecules that were distributed within this thin section of lysosomes. So the instrument that is used to perform this super-resolution imaging is actually quite simple, and this is one reason why many researchers are excited, because they don't need much money in order to do this type of experiment. So you need a cold CCD camera to image the individual photons that are being emitted by these photoactivatable fluorescent proteins. You also need a variety of different lasers for activating as well as imaging the photoactivatable fluorescent proteins. And finally, you need a total internal fluorescence microscopic objective in order to be able to limit the sampling area of your specimen to only about 100 nanometers off the cover slip. We typically use this because if you just use a conventional objective, the fluorescence from out of focus of molecules greatly increases the background photon, making it much more difficult to precisely map the distribution of individual molecules. Now here's an example of the type of resolution that you can get using PALM super resolution imaging. To the right is an aggregate of 50 nanometer beads that have been labeled with a photoconvertible protein chiate. To the left is the PALM super resolution image. And if we zoom up on that image, you can see that with this technique, you can decipher the distribution of these individual 50 nanometer beads. Now, of course, many people want to move into cells, and it's possible to do this type of super resolution imaging in fixed cells that have been plated on a cover slip. Now, I mentioned we use total internal reflection microscopy to limit the imaging to just about 100 nanometers off that cover slip in order to avoid photons that are being emitted by your probe in other places in the cell, and hence could interfere with the signal that you're getting of the single molecules that are of interest in this plane right here. So just to give you some examples of what you can get with this approach, here are focal adhesions tagged with a photoconvertible protein, tandem dimer EOS, and if we zoom up on this, you can see, and I'll move over here, the localization of these individual vinclin molecules in these focal adhesions that are found at the base of cells. And these focal adhesions are sort of like the feet of cells that allow cells to crawl. This is another example of a EOS molecule tagged to an ER resident protein, the Reticulum 1. And if we zoom up on one element of the endoplasmic reticulum, you can see how these molecules really outline the surface, the individual tubular elements that comprise this dense network of the ER. Now, many organelles within cells are not localized right within the 100 nanometer turf zone of a cell that's put down on a cover slip. And so there are various strategies one can use to visualize intracellular compartments using the super resolution approach. And one way is to fix cells and section them as if for electron microscopy. Here, this cell has been sectioned, this is about a 100 nanometer section that's going to then be placed on a cover slip and imaged in that turf zone. So this is a slice of a cell that's expressing a photoactivatable fluorescent protein, and we're going to look at what the organelles within that slice, how molecules within those organelles distribute. So here's one slice through a cell expressing a mitochondrial matrix protein tagged with the photoconvertible protein tandem dimer EOS. Now, the advantage of creating a thin section through the cell and doing PALM on that thin section is that after you've imaged the single molecule distribution within that sample, you can then take the sample and put it in the electron microscope to do transmission EM imaging in order to compare the ultrastructure of the structure of interest to the distribution of the molecules. And this is shown here for the mitochondria. So this is the transmission EM of a mitochondria, and this is the distribution of the mitochondrial matrix protein within that. And you can see that in this overlay here, that they very nicely overlay. And what this gives you is a high-density alternative to conventional immunogold labeling approaches, which typically in a specimen like this where you're using antibodies tagged with immunogold would give you maybe 10 or 20 gold particles to define the distribution of your protein. Here we're looking at 5,500 GFP, the photoactivatable fluorescent protein tags molecules that have been resolved using the PALM technology. Now there are a variety of photoactivatable fluorescent protein classes that have emerged over the last 4 years. The initial one was the photoactivatable GFP, which switches from dark to green and which has been used in a number of different techniques. But more recently, there's been a lot of excitement about these photoconvertible proteins that start off green, but in response to UV illumination, change their emission spectrum so that now they fluoresce red and give off a red signal. There's also reversible photoconverters that can go from green to red and then back from red to green. So all of these classes of different photoactivatable or photoswitchable fluorescent protein allow us now to use them for doing two-color imaging using this super-resolution approach. And here's an example where the photoactivatable GFP tagged to clathrin is being expressed with a photoactivatable M-cherry tagged to transferrin receptor. Now what this allows is the simultaneous detection of these two molecules, transferrin receptor and clathrin, in a single cell, simply by switching on with a UV laser pulse either the M-cherry, which gives you the red signal, or the PA-GFP, which gives you the green signal. And what you can see in this super resolution image right down here is a co-distribution of these two molecules within densely packed structures that we believe are clathrin-coated pits. The clathrin-coated pits are known to be defined by the activity of this clathrin coat protein, and the transferrin receptor, after binding transferrin, is known to be endocytosed within these clathrin-coated pits. And we think that's what is being seen here in this super resolution imaging approach, is the co-distribution of these molecules in these coated pits. Now, up to now, I've showed examples of using this single molecule localization approach called PALM for looking at specimens pretty much in 2D, where we're putting them on a microscope, on a cover slip and looking in turf. But many questions that are important for cell biology to address require 3D information in terms of for a global view of how these molecules are arranged over 3D within cells. And a very exciting new advance with PALM has come with this technique called Interferometric PALM, or iPALM. And in iPALM essentially couples an interferometric strategy to PALM imaging. So in this little QuickTime movie here, this illustrates the basic principle of iPALM. Essentially, a specimen is being imaged by two different objectives. And if you zoom up in here, what you can see is an individual molecule, photoactivated molecule, that's giving off a photon. Now that photon distributes as a wave in 360 degrees, and it's going to move into either the top objective or the bottom objective, and then track through that objective into this three-way beam splitter. Now, depending on where the photon emitter is positioned when it gives off its photon, the waves from that photon, when they recombine in this three-way beam splitter, will either constructively or destructively interfere with each other. And that can be sensed by these three CCD cameras to determine with nanometric accuracy where this photon has been emitted in Z space. So this is the principle behind iPALM, and it gives remarkable 3D resolution. So this is a resolution comparison of confocal microscopy in Z and XY compared to PALM, where you get incredible improvement in your XY and improvement in Z based on the TIRF using total internal reflection microscopy. What iPALM gives you is an additional improvement in Z, such that you now have an accuracy of about 10 nanometer in this Z dimension in terms of where a particular photon emitted by a molecule is positioned. So here's an example of iPALM imaging of VSV-G protein on the plasma membrane. Now, what you're looking at, all these little dots represent single molecules of VSV-G that are localized on the plasma membrane. Now, each of these molecules are different, have different colors associated with them based on how far up from the cover slip the molecule is positioned. And this is a lookup table showing from 0 to 225 nanometer where these molecules are distributed in Z space. So if we take this little section, this little rectangular area right here and rotate it on its side, you can see in the side view the surface of the plasma membrane. You can see the dorsal as well as the ventral surface of the plasma membrane, which in this case is less than 200 nanometers apart. So this is an extremely exciting approach because it allows you for the first time to get a three-dimensional perspective on the distribution of organelles and membrane systems within cells. So here's another example. This is iPALM imaging of the alpha V integrin that makes up the focal adhesion, which forms these little feet-like structures at the bottom of cells. Now these feet-like structures are shown in are color-coded yellow here only because they're positioned very close to the plasma membrane. What you're looking at here is the distribution of thousands of molecules that have been detected using PALM and positioned in Z based on this interferometric approach and color coding according to their Z position. Now what's particularly interesting is that you can see that different positions of the molecule in Z have very different spatial organizations. So for instance, the molecules that are color-coded purple and blue are all found in structures that have this tubular reticular pattern. The reason is that these molecules are within the endoplasmic reticulum, which is positioned at about 225 nanometers off the surface of cells. The molecules that are color-coded yellow are simply the ones positioned close to the cover slip, and you can see all of those molecules are associated in structures that have the characteristic morphology of focal adhesions. So if we take all of this information, we can get a 3D perspective on the distribution of this molecule integrin as it passes through the entire secretory pathway. We can see it in the ER, on the plasma membrane, and in these focal adhesions, and we can get a super-resolution perspective on how all of these membranes are organized within cells. Now, up to now, I've talked about how you can use PALM in fixed specimens, but you can also use these single molecule super-resolution approaches in live cells, and that's illustrated here, in order to track the movement of individual molecules. Now, I showed in the second part of this series how using photoactivation or photobleaching, you could easily look at the diffusional behavior of molecules using diffraction-limited imaging. But that approach involves imaging large numbers of molecules, large populations. With single particle tracking PALM, you can look at the behavior of individual molecules and determine whether an individual molecule is freely diffusing, undergoing some type of active transport as a result of the activity of a motor protein, or undergoing some type of confinement. And to illustrate that, here we have the VSV-G protein again at the cell surface, and what you're looking at is the vectors, the tracks from these molecules that have been detected over a period of the lifetime of these photoactivatable fluorescent proteins. And if you just look right here, this is just a zoom up showing the tracks of individual VSVG molecules. Each one of these different color lines represents the track taken by an individual VSVG molecule at the cell surface. Now, with these tracks, you can do a lot of things. So, for instance, here we have the tracks have been translated into a diffusion coefficient. So each one of these little spots represents the diffusion coefficient of an individual molecule that originated at that spot. And this lookup table here just shows what that diffusion coefficient was. Now, what you can see from this is that there are subsets of these molecules, in this case VSVG, that are highly mobile, and then there are subsets that are less mobile. And this gives you a spatial sense of where those molecules are distributed, ones that move fast versus slow, across the surface of the cell. Now, as a comparison, this is the HIV gag protein, which has been mapped. The HIV gag protein is a key component of the HIV virus that is responsible for AIDS. And when you express a photoactivatable form of this virus, you can then map its behavior on the cell surface, a photoactivatable form of the HIV gag protein that forms the coat of this virus. You then can map its distribution on the cell surface of the cell, and what you see is that these molecules, by and large, are not moving as rapidly as the VSVG. Large numbers of these molecules have very low diffusion coefficients, as illustrated in this lookup table here. And many of the molecules are clustered in these aggregates. We think these clusters represent sites of viral budding on the surface of the cell, and we're hoping that using this technique, we can get insight into how the virus budding process occurs vis-a-vis the assembly of these proteins that form the coat of this virus. Now finally, with the single particle tracking PALM, where you're looking in living cells at the behavior of single molecules, you can go off the cell surface and into the cell to look at the behavior of cytoskeletal components. And here's an example where we've been imaging actin filaments using a photoactivatable fluorescently tagged actin molecule. So this is a living cell, and you're just tracking the movements of individual actin molecules. Because the actin molecules that are part of filaments are moving relatively slow compared to actin that's free in the cytoplasm, you can actually track the behavior of individual actin molecule within that filament. And this is just a zoom up of a portion of the periphery of the cell showing the behavior of the actin molecules there. And you can see that all of these molecules are moving, and they're moving in a directed fashion, a fashion where the molecules are moving inward along tracks of actin filaments that are undergoing retrograde actin flow. And using this approach, we and others are finding some new aspects of this movement because of the ability to image single molecules. So what all this means is that there are tremendous possibilities available for researchers now for addressing questions that really have been off limits up to now because they involve imaging individual molecules. So with these techniques that involve single molecule localization like PALM, you have now the ability to go in and map out distributions of molecules within nanometric structures, as well as the dynamic movements of these molecules through this type of tracking.